Imagine a geneticist sitting in a laboratory somewhere in Europe, staring at a sequenced genome on a screen. The genome belongs to a person who died approximately 10,000 years ago, whose skeletal remains were found in a cave in what is now Spain. The geneticist is looking at the genes associated with skin pigmentation, and what they are seeing is producing the kind of cognitive dissonance that good science occasionally delivers when the evidence contradicts the assumption. The person whose genome is on that screen lived in Western Europe. Their ancestors had been in Europe for thousands of years. By any geographical definition, this was a European human being, and yet the pigmentation genes tell a story that upends the intuitive assumption about what ancient Europeans looked like. An assumption so deeply embedded in popular imagination that even professional researchers initially found the results surprising enough to double-check. This person almost certainly had dark skin. Dark eyes. The combination of features that modern European populations associate with people from entirely different parts of the world. The blue eyes and light skin that many people mentally project onto ancient Europeans, onto the hunters who painted the cave walls at Altamira, onto the people who built megalithic monuments across the Atlantic coast, were, according to the genetic evidence, largely absent from European populations until surprisingly recently. The pale complexion that some people treat as definitionally European, as an ancient and fundamental characteristic of people from that part of the world, turns out to be a relatively recent evolutionary development with a traceable origin, identifiable mechanisms, and a timeline that most people find genuinely unexpected. The question of when the first light-skinned humans appeared, in Europe, and more broadly across the populations where pale complexions are common today, has a scientific answer. It is not the answer that historical mythology, racial ideology, or intuitive assumption would suggest. It is considerably more recent, considerably more complex, and considerably more interesting than any of those frameworks predicted. Before addressing the timeline, the biology of skin pigmentation needs to be understood clearly, because the mechanism determines why the variation exists at all and why it evolved when and where it did. Skin color in humans is determined primarily by melanin, a pigment produced by specialized cells called melanocytes distributed through the skin. Melanocytes in all humans exist in roughly similar numbers regardless of skin color. What differs is the type and quantity of melanin they produce. Eumelanin, the dark brown to black pigment, provides stronger UV protection. Pheomelanin, a lighter, reddish-yellow pigment, provides less protection. People with darker skin have melanocytes that produce more eumelanin in larger, more stable packages. People with lighter skin have melanocytes producing less eumelanin and more pheomelanin, and the melanin packages are smaller and break down more readily. This melanin variation is controlled by numerous genes. The human pigmentation system is polygenic, meaning multiple genes contribute to the outcome, which is why skin color in human populations shows a continuous gradient of variation rather than discrete categories. The major genes involved include MC1R, SLC2, 4A5, SLC45A2, HERC2, OCA2, KITLG, and several others, each contributing to various aspects of pigmentation through different biological mechanisms. Changes in any combination of these genes can shift skin tone lighter or darker. The key question for understanding the evolution of skin color is not what genes are involved, we know those, but what selection pressures drove the variants associated with lighter skin to become common in particular populations. And that question has an answer rooted in vitamin D synthesis and ultraviolet radiation that is now well established in the scientific literature. The evolutionary framework for understanding human skin color variation begins with our origins in equatorial Africa, where abundant UV radiation created strong selection pressure for dark, eumelanin-rich skin as protection against UV damage. UV radiation has multiple effects on human biology, some beneficial and some harmful. On the harmful side, UV radiation damages DNA, causes sunburn, and, with chronic high exposure, dramatically increases skin cancer risk. UV radiation also destroys folate, a B vitamin essential for DNA synthesis and critically important during pregnancy for neural tube development. Populations living under high UV conditions face strong selection pressure for skin that limits UV penetration, because the individuals with better UV protection suffered less folate depletion, fewer UV-related health complications, and better reproductive outcomes. On the beneficial side, UV radiation is required for the synthesis of vitamin D in the skin. Specifically, UVB radiation converts a cholesterol precursor in the skin to pre-vitamin D3, which is then converted to active vitamin D through subsequent steps. Vitamin D is essential for calcium absorption and bone metabolism, immune function, and various other physiological processes. Deficiency causes rickets in children and osteomalacia in adults, both of which impair physical function and reproductive success. In populations living at low UV levels, high latitudes, particularly during winter months, skin that permits greater UV penetration for vitamin D synthesis provides a survival advantage. The relationship between UV levels and skin color across indigenous human populations is remarkably consistent. Populations living near the equator tend to have darker skin, populations living at higher latitudes tend to have lighter skin, and the gradient follows UV availability with a precision that argues strongly for UV-related selection as the primary driver. This relationship was first systematically documented by biological anthropologist Nina Jablonski, whose work establishing the UV skin color correlation across global populations represents one of the clearest demonstrations of natural selection's operation in human populations. The timeline of modern human migration out of Africa and into Eurasia is the necessary backdrop for understanding when lighter skin evolved, because lighter skin variants only become advantageous after populations move into lower UV environments. Modern Homo sapiens emerged in Africa approximately 300,000 years ago, with our species' ancestors carrying the dark skin appropriate to equatorial UV levels. The migration out of Africa, the dispersal that eventually populated the rest of the world, began in earnest approximately 60,000 to 70,000 years ago, with populations spreading into the Middle East, then across southern Asia, eventually into East Asia and Europe, and later into the Americas and Oceania. The populations moving into higher latitude environments immediately faced a different UV regime. Northern Europe, particularly in winter, receives dramatically less UVB radiation than equatorial Africa. At latitudes above approximately 50 degrees north, UVB levels during winter months are so low that essentially no vitamin D synthesis occurs in human skin, regardless of its pigmentation. Even during summer, UV levels at northern European latitudes are dramatically lower than at the equator. A population arriving in northern Europe from Africa with dark, UV-blocking skin faced a genuine vitamin D synthesis challenge, particularly during the long northern winters. This challenge created selection pressure for lighter skin variants that permitted greater UV penetration. But, and this is crucial for understanding the timeline, selection pressure takes time to shift allele frequencies in a population. If the selection advantage of lighter skin variants in a northern European environment was strong but not overwhelming, the genetic change would occur over hundreds of generations. If the selection was strong, the change could occur more rapidly. The actual timeline, as revealed by ancient DNA analysis, tells us something important about both the strength of the selection pressure and the source of the variants that eventually became common. The ancient DNA evidence is where the timeline becomes both precise and surprising, and where the assumptions embedded in popular imagination most thoroughly collide with what the evidence actually shows. Ancient genomic analysis has advanced dramatically since the first ancient human genomes were sequenced in the early 2000s. Researchers can now analyze the pigmentation genes of ancient humans from skeletal remains across a wide geographic and temporal range, providing a direct window into what past populations actually looked like rather than what we assume they looked like. The results have been consistently surprising to non-specialists and have required significant revision of assumptions about the pace and nature of pigmentation evolution in European and other populations. The Mesolithic hunter-gatherers of Western Europe, the people who lived in Europe from approximately 40,000 years ago after the first modern human arrival, through the end of the Ice Age and into the early Holocene, show consistent dark skin pigmentation variants in their genomes. The famous Cheddar Man, whose skeletal remains found in Somerset, England, date to approximately 10,000 years ago. By the way, if you want to go even deeper into the secret lives of ancient humans, I just launched a full course called The Secret Lives of Ancient Humans. 10 modules, 3+ hours of content covering everything we never talk about on YouTube. What they feared, how they communicated, what hunted them, and much more. Plus an exclusive PDF guide included. Link in the description. See you inside. Was analyzed using ancient DNA methods in 2018, and the results showed high probability of dark to dark brown skin, blue eyes, and dark curly hair. His appearance, reconstructed from genetic data, looked markedly different from the pale-skinned Northern European populations that would later inhabit the same territory. Cheddar Man was not alone. Mesolithic hunter-gatherers from across Western and Central Europe, from Spain, from Luxembourg, from Hungary, from Scandinavia, show similar pigmentation profiles in their ancient genomes. The pale skin that modern Northwestern Europeans carry was largely absent from the people who hunted mammoths, buried their dead in ochre, and built the earliest post-glacial cultures of Europe. These were dark-skinned Europeans. A phrase that sounds paradoxical to people who have absorbed the cultural equation of European ancestry with pale complexion, but which is simply what the genetic evidence shows. So when did lighter skin actually become common in European populations? The answer involves not just gradual evolution of variants already present in the hunter-gatherer population, but a dramatic demographic transformation that brought new genetic variants into Europe from outside. The Neolithic agricultural revolution, which began in the Middle East approximately 10,000 to 12,000 years ago, involved the spread of farming populations out of Anatolia and the Fertile Crescent into Europe beginning around 9,000 years ago. These Anatolian farmers carried a different set of genetic variants from the European hunter-gatherers they encountered and, over centuries, largely replaced demographically. Crucially, the Anatolian farmers carried some lighter skin variants, particularly variants of the SLC24A5 gene that were present in their population, and that provided a vitamin D synthesis advantage in the European environment. The SLC24A5 variant associated with lighter skin in European populations appears to have originated and been rising in frequency in populations of the Middle East and Anatolia during the period immediately before the Neolithic expansion into Europe. Its presence in the Anatolian farming populations meant that as those populations spread westward into Europe, they carried this lighter skin variant into regions where it provided a strong selective advantage. But even the Anatolian farmers were not pale-skinned in the way modern Northwestern Europeans are. Analysis of Neolithic European genomes shows that while the SLC24A5 light skin variant was present and increasing in frequency, other pigmentation genes associated with lighter skin in modern populations were still at relatively low frequencies. The Neolithic farmers of Europe appear to have been intermediate in skin tone, darker than modern Northern Europeans, but lighter than the Mesolithic hunter-gatherers they were replacing and assimilating. The third major population movement that shaped modern European genetics, and with it, modern European pigmentation, arrived from the east approximately 5,000 years ago in the form of Yamnaya or steppe pastoralists. These people, whose genetic ancestry traces to the Pontic-Caspian steppe north of the Black and Caspian Seas, expanded westward into Europe during the late Neolithic and Bronze Age in a migration that left a major genetic footprint across much of northern and western Europe. The steppe pastoralists carried yet another set of pigmentation variants, and the genetic mixture produced by the combination of Mesolithic hunter-gatherer ancestry, Anatolian farmer ancestry, and steppe pastoralist ancestry, in proportions that varied across different regions of Europe, produced the range of pigmentation profiles that characterize modern European populations. The complete package of lighter skin variants associated with modern Northern European pale complexion, involving multiple genes including SLC24A5, SLC45A2, HERC2/OCA2, and others, appears to have reached high frequencies in European populations primarily within the last 6,000 to 8,000 years, with some variants still increasing in frequency as recently as 4,000 to 5,000 years ago. Some analyses suggest that the combination of lighter skin variants that characterizes many modern Northern Europeans may not have been common until the Bronze Age, a period that feels historically recent given that agriculture, writing, and early civilization had already emerged by this point. This timeline means that the people who built Stonehenge may have had meaningfully different pigmentation from the people who would later inhabit the same landscape. The people who created the first written records in Mesopotamia were contemporaries of European populations still carrying darker skin variants. The pale complexion associated with Northern European populations emerged at the population level, later than the agricultural revolution, later than the Bronze Age transition in some regions, and through a combination of migration, population replacement, and natural selection operating on variants that arrived in Europe from multiple source populations. The vitamin D synthesis advantage provides the strongest explanation for why lighter skin variants were positively selected in high-latitude populations, but there is ongoing scientific discussion about whether additional selection pressures contributed to the speed of the change. Folate protection, the advantage of darker skin in blocking UV radiation that destroys folate, operates in reverse at high latitudes. If folate destruction is less of a problem at low UV levels, the protective advantage of dark skin is reduced, which in effect reduces the cost of having lighter skin variants rather than providing a positive advantage for lighter skin. This dynamic, reduced disadvantage rather than increased advantage, may have contributed to allowing lighter skin variants to drift upward in frequency even without strong positive selection at the earliest stages of high-latitude occupation. Sexual selection has been proposed as an additional factor, with the argument that pigmentation differences may have influenced mate choice in ways that accelerated the spread of lighter skin variants beyond what vitamin D selection alone would predict. This hypothesis is difficult to test and more controversial than the vitamin D synthesis explanation, but it remains in the literature as a candidate contributing factor. The relative speed of the pigmentation change in European populations, faster than purely gradual drift would predict, but with timing that suggests multiple input events rather than continuous gradual change, is most consistent with the combination of migration, bringing new variants into Europe, and strong natural selection, rapidly increasing their frequency once they were present. The vitamin D synthesis selection pressure at high latitudes was strong enough that once lighter skin variants were introduced through migration from populations where those variants were already present, they rose in frequency relatively rapidly. The comparison with East Asian populations adds an important dimension to the global picture of lighter skin evolution, because East Asian populations, who also live at high latitudes and carry lighter skin than equatorial African populations, developed their lighter pigmentation through largely different genetic variants than Europeans. If lighter skin in high-latitude populations had evolved only once and spread from a single ancestral population, East Asian and European lighter skin would be the product of the same genetic variants. Instead, the genetic analysis shows that the lighter skin of East Asian populations involves different gene variants, particularly in the MC1R gene and others, from those that predominate in European populations. The lighter skin of East Asians and the lighter skin of Europeans are the result of convergent evolution, different genetic solutions to the same adaptive problem in different populations. This convergent evolution provides some of the strongest evidence that natural selection for vitamin D synthesis at high latitudes is the genuine driver of lighter skin evolution in both populations, rather than the lighter pigmentation being an incidental feature of some shared ancestral population. When selection pressure is strong enough, different populations find different genetic solutions to the same problem, and the result, lighter skin suited to lower UV environments, looks similar despite the different underlying genetic architecture. The timeline for East Asian pigmentation lightening shows similar patterns to the European case. Lighter skin variants are not ancient in East Asian populations but appear to have increased in frequency over the last several thousand to tens of thousands of years as populations moved into and adapted to higher latitude environments. What does all of this mean for understanding human diversity, and why does the answer matter beyond the purely academic? The genetic history of human pigmentation reveals skin color as exactly what evolutionary biology would predict it to be: a relatively recent, highly variable, geographically distributed adaptation to UV conditions that has changed repeatedly across human history as populations moved between environments, mixed with other populations, and responded to local selection pressures. It is not ancient, not fixed, not a deep marker of ancestry in the way popular imagination often treats it. The Europeans of 10,000 years ago were dark-skinned. The pale skin of modern northern Europeans is younger than agriculture. Possibly younger than Stonehenge. Certainly younger than the cave paintings of Altamira and Lascaux, whose creators we now know were dark-skinned inhabitants of Ice Age Europe. The people who hunted reindeer in Scandinavia during the early Holocene were dark-skinned people who had adapted to other challenges of that environment without yet showing the pigmentation shift that their descendant populations would later undergo. This is not a peripheral detail of human biology. It is a central illustration of how natural selection operates on visible traits over timescales that are short in evolutionary terms, but still extend far beyond any historical period, and of how dramatically populations can change in appearance through migration, admixture, and selection without any change in the fundamental nature of the organisms involved. Skin color is a recent adaptation to local UV conditions, implemented through multiple genetic pathways, distributed across populations in proportion to latitude and UV exposure with remarkable consistency, and evolved at different times in different populations through different genetic mechanisms. The first humans with the pale skin profile characteristic of modern northern Europeans appeared approximately 6,000 to 8,000 years ago, possibly more recently, as the result of a combination of migrations and natural selection operating on variants that arrived in Europe from populations that had already started developing lighter skin in response to similar UV pressures in the Middle East and Central Asia. That person, the first Northern European with what we would recognize as Northern European pigmentation, was living during a period of enormous cultural and demographic change, part of populations that had recently arrived in Europe or that had mixed with arriving populations, navigating environments that required constant physiological adaptation. They had no concept of race, no framework within which their skin tone carried the social meaning that later human societies would assign it, and no awareness of the genetic change that had produced their appearance as the outcome of a straightforward evolutionary process, responding to the amount of ultraviolet radiation hitting their skin on an average day. They were just people, adapted to their latitude, doing what adapted populations do, surviving, reproducing, and leaving genetic descendants who would eventually carry their pigmentation variants across the modern world, while entirely forgetting that those variants were once novel, once rare, and once absent from a Europe full of dark-skinned humans navigating the same cold northern landscapes under the same limited northern light.